Welcome, everybody. It's a pleasure to have you here. And again, on behalf of the Global Institute for Water Security and the Global Water Futures program, who underwrite this lecture series, I'm thanking you and thanking them. A little bit of housekeeping. Next week, we will not have a presentation. We'll have a down week. The week after, Yin Van Reinfelder from Rutgers University will be here to talk about global groundwater. But today I'm really happy to introduce Kathleen Weathers. Kathy is a senior scientist at the Cary Institute of Ecosystem Studies in Millbrook, New York. She's worked there in one form or another for, I think, 30 years or thereabouts. She's also on the graduate faculty at Rutgers University. She's a visiting scholar at Dartmouth College, and she has a faculty appointment at Cornell in the program in biogeochemistry and environmental change, where she works closely with grad students. Kathy's really served as a thought leader in ecosystem science for many years and decades. She's an elected fellow of the American Association of the Advancement of Science. She's Mary Tharp Visiting Fellow, or was at Columbia University. She's received NSF's Director Award, focused on the water sustainability and climate program, something I had the pleasure of working on, and that Kathy was really the brainchild of. She served as a rotator at NSF, kind of a program manager for a period some years ago. She's also been very active in community leadership. She's chair of the Gordon Research Conference. As many of you know, that's really the signature conference in this area of catchment science. She's co-chair of GLEON, something you'll hear about, no doubt, in today's seminar, and she served on a number of advisory boards, really key strategic advisory boards. A couple I'll mention, the EPA's Clean Air Scientific Advisory Committee, and of course she serves on the Board of Trustees, or did, with the Hubbard Brook Research Foundation, where she's worked for many, many years. She served as editorial board member and advisory board for journals like Tree Physiology and Biologist. And it's just terrific to have her here in Saskatoon today. We've had a great discussion with the students this afternoon on some of her FOG work. And today we're going to hear more about her lake work. And I think this touches on many things that folks at the Institute, Environment Canada, and across the university are working on. So, Kathy.
Thank you. It's been a delight to be here, and I'm really honored to be here as part of this program. And I'm excited to talk to you about an area of research that, you know, in 35 years, it's only been the last 10 or 12 years that I've been involved in. I was trained by a limnologist, but not as a limnologist. So today I'm going to give you just an overview of some of the research that we've been involved in particular lakes, and then scale up to the globe and talk a little bit about GLEON, the Global Lake Ecological Observatory Network, how we operate, because I think that's really important, and some of the results that we've gotten. So this is kind of interesting, having two screens, two groups of people. I like to see you, so I'll probably be wandering back and forth. So before I get started, I want to acknowledge my collaborators. All of what I'm going to say today is a result of collaborative research, much of it with my colleagues who work in New Hampshire and Maine with me, Kathy Cottingham, Cary, Holly Ewing, Meredith Greer, and then there are a number of people with the GLEON network, and really importantly, for this last decade of my research, a number of citizen scientists and citizen science groups. And that's one of my messages today, is there are ways in which we can think about doing science a little bit differently in the future, I think, democratizing science, and I hope to illustrate some of those. And I am not averse to someone asking questions in the middle of this if there's anything that's unclear. Okay, so here's my roadmap. The first half to 3/4, let's see, I can't see that from here. I want to talk specifically about a research project that we've been working on from many different aspects for the last 10 or 12 years. And then I want to talk, as I said, I want to scale up and talk about this intersection of sensors and citizen scientists and cyanobacteria, and talk about the global network. So I knew nothing about citizen science and citizens and science until about 12 years ago when I went on sabbatical leave, which I'll explain in just a minute. At least in the US, I'm not sure about Canada. In the US, the citizen science and citizens and science has really come to the fore. at the federal level, at the local level, and so there's a lot of conversation. And even within GLEON, which I'll talk about later, there are all kinds of citizen science projects that are showing up. This is one that showed up really recently, high-profile journal using citizen science research. So, where my world intersected with lakes, other than from a personal standpoint, was about 12 years ago, where I did a crazy sabbatical leave. All my colleagues said, you know, if you go on a sabbatical leave, go work with Jeff, learn something about isotopes, go somewhere else. And I got talked into going to a lake association and working with that lake association to understand how science might underpin their outreach and education. I had spent a lot of time at this lake, Truth in Advertising. I've been there ever since I was about 1 year old. My family spent time on that lake, but I'd never taken my scientific eye and looked at the lake. So literally, here I am diving into the lake a lot of years ago, and I dove into something new about 12 years ago. So to orient us, this is Lake Sunapee, New Hampshire. It's a deep lake. It's a clear lake. Its watershed is probably not unlike a lot of at least eastern Canadian lakes, where it's mostly forested, except for right around the edge of the lake, where there's about 6% transportation. This lake association is a really forward-looking lake association. They've been around for 120 years. They're the oldest environmental organization in New Hampshire. They had operated with volunteers up until about 20 years ago, and now they have a paid staff who do outreach and education. They reach, and these are towns surrounding this lake of maybe 3,000 people or so, but they reach 4,000 to 5,000 kids and adults a year with their outreach and education program. And their focus really is on not only the lake, but the region. And they're very interested in developing programs and exporting them to other lake association, other groups at no charge. They are kind of thought leaders and action leaders. So again, this is a clear water lake. This is an oligotrophic lake, nutrient-poor lake. It's deep, as I said. There are long-term water quality data from this lake, and actually many lakes in the US and especially in the eastern United States for over 25 years, citizens collecting samples and having data or samples analyzed. It's also a drinking water source for residents, and it has a developing shoreline. So in 2004, when I arrived for my sabbatical leave, this had just happened in this Clearwater lake. This is enter Gleotrichia. I'm going to refer to them as Gloeo, a cyanobacterium. And this cyanobacterium was discovered by the citizens who live around the lake, who use the lake. We as scientists never would have noticed because we weren't spending time there. So they discovered it. They brought it to the attention of me when I was on sabbatical, and I brought it to the attention of students and colleagues who were in the area. I should also say that before my sabbatical, I think there was only one scientific paper on Lake Sunapee, and that was about Lake Sunapee trout, which is now extirpated, was an endemic trout. So not a lot of research had gone on there. And when I went on sabbatical, I said, well, there are a couple of ways in which you can know more about your lake. One is if you want to solve problems, you hire a consulting firm and solve those problems. But if you want to know more from scientifically, you need to engage people in doing research at the lake, or you need to engage the research community to interpret data and science from lakes and see how it's applied to Lake Sunapee. And all those things have happened since then. I'm not going to tell that part of the story, but we were successful in engaging all kinds of people to do research on Lake Sunapee now 10 years later. So again, this is an oligotrophic lake. It's iconic. It's a source of drinking water, fabulous recreation. And the first thing that I and colleagues were asking was, okay, eutrophication, you get cyanobacterial blooms, but what are cyanobacteria doing blooming in this lake? And so I want to take you through some of our hypotheses and then some of the data and then some of the what next. So I probably don't need to give this group background, but I'm going to do it anyway. You know, I'm sure, that eutrophication, nutrient enrichment, is a global problem. It's happening all over the world. It's happening at higher frequency, as far as anyone can tell. And these cyanobacterial blooms have a lot of negative impacts, from aesthetics. ecosystem services, economic value. There are some economic models that are linked to water quality in lakes and housing prices. There are biogeochemical impacts, which piques my interest, that where decomposition, you get a decrease in oxygen availability, so-called dead zones, fish kills, other issues. So there are a whole suite of issues. And many of these cyanobacteria are toxic, not all, but many are. Their health effects seen on dogs, livestock, wildlife, people. And some of the better studied forms are actually neurotoxins and hepatotoxins. So it's a serious potential health issue, these cyanobacteria. As a matter of fact, in the news, actually this year, this one is from last summer, on our national public radio station, this was one of the headlines. Poisonous algae blooms threaten people and ecosystems across the US. So, again, of concern. So what do we know about eutrophication and cyanobacterial blooms, or what did we know at that point? Well, they've been definitively linked in the literature, maybe by some of the literature that you all have written, to phosphorus availability, a nutrient. There's increasing evidence, although there's a huge debate, increasing evidence that increased availability of nitrogen is also an issue in eutrophication and cyanobacterial blooms. So here's the conventional wisdom that we went into this question with, which is that phosphorus and nitrogen increase in the water body as a result of human activities, agriculture, from waste, and you get cyanobacterial blooms. So it's the nutrient enhancement that drives the blooms, and that's what we see in a number of different places. But again, what about these clear water lakes? There aren't high nutrients in these clear water lakes. Yet this is our Gloeo, which is quite large actually, is showing up. So again, this is the ecological puzzle in oligotrophic systems that we've been chasing after for a number of years. So again, a little bit of background. Sediments are the major store of phosphorus in this lake and many lakes that are surrounded by forests and not by agriculture. It's been referred to as the phosphoric time bomb. So imagine these huge stores of phosphorus in the sediment of lakes. If you look up, so in the atmosphere, there's this huge resource of nitrogen, but it's chemically bound, so most organisms can't utilize that nitrogen. So we have been hypothesizing that there's this Pandora's box issue from Greek mythology, where every trouble known to humanity was in Pandora's box, and when opened, ended up in the system. And our question continues to be, are cyanobacteria, in some cases, special cyanobacteria, are they biotic keys to Pandora's box, able to unlock some of these ills of the world? And how would that happen? Well, you likely know that many cyanobacteria can fix nitrogen, so they can unlock the atmospheric pool. And conventional wisdom would say that really in the phosphorus pool in the bottom of lakes, it's a geochemical response, so anoxia. Lack of oxygen allows phosphorus to be released, and that's the mechanism by which this phosphoric time bomb is set off. But what we're learning is that benthic, bottom water cyanobacteria, can enhance the phosphorus released from sediments. So they overwinter on the sediment, and these germinating cyanobacteria can access the sediment and transport it upwards. So it would look something like this. And those cyanobacteria can also sink and access available phosphorus and then rise back up to the surface. So the potential exists for these cyanobacteria to be keys. So the hypothesis that we've been working on and getting some pushback on is that these cyanobacteria may be leading eutrophication. They're driving the train as opposed to riding the train, where they are packets of phosphorus and nitrogen that can be lysed, that can end up in the internal cycling of the system and result in enhanced phosphorus and nitrogen oligotrophic or eutrophication that follows the cyanobacteria. So that's kind of the question. And I'm going to show you now some of the data that have led us to not let go of this hypothesis yet. So here, actually, this is drinking water from Lewiston, Maine, that was collected by our colleague, Holly Ewing, from the drinking water supply of Lewiston, Maine, with all these cyanobacteria in it. And it's, again, it's a potential problem that is not being filtered at the moment. Okay, so what about the data? So again, I started sabbatical 2004. Cary, a graduate student then, who's now at Virginia Tech in 2005, showed up on my doorstep, said, I'm interested in a research project, an undergraduate research project. And I said, what are you interested in? And she said, oh, I love algae. I said, you love algae? Any algae? We've got a problem for you. So she became engaged in this project and got a dissertation and now has her graduate students studying this issue. So this is what the colonies at maximum bloom looked like in 2005, 2006, and then 2007. We were able to capitalize on this jump, which was a little bit startling, at least to people who were looking at the cyanobacteria, and get some National Science Foundation funding, which is called rapid funding, to deal with issues that have popped up and it looks like they're going to cause trouble. So that began our funding cycle. Then, of course, the next year, thankfully we had the funding. The next year, the blooms weren't as large. And this, I don't have 2016 and 2017 in here yet, but this is what the maximum number of colonies, or number of colonies at maximum bloom have looked like over the years. So, heterogeneity, it's an issue. Again, it poses an interesting ecological puzzle, but you know, there's high variability in the blooms in ways that we might not predict. And you can also see that at the maximum or at the high bloom years, it's kind of on its way up. But again, heterogeneity is key here. Well, we also wanted to know and had heard through the grapevine whether this was unique to the Lake Sunapee region. And it turns out it's not. So Cary, again, as a graduate student, went to a number of lakes in Maine, in New Hampshire, and in New York. And I understand, actually, I'm not sure it's Gloeotrichia, but there are all kinds of blooms that are happening in similar sorts of situations in eastern Canada as well. And almost all the places where we looked, there were Gloeotrichia. So this is happening. It's happening across, excuse me, across the region. So what's one of the first questions you ask is it new? Did it come in on a boat? Is it new or is it old? And so with the leadership of Holly Ewing, our colleague at Bates College, cored a number of lakes and found that Gleo is actually not new. This is more confusing than not. So I'm showing data from a core from Lake Sunapee in New Hampshire and showing you that at the top, can you see that at all? At the top of the graph is the most recent time, and then it goes down. You can see on the y-axis it goes down. And what I'm showing you is the presence of Gloeotrichia in the sediment. So what you can see immediately is that there have been times, actually earlier than the 1853, and some European colonization where there's been more than there is today. So that's really confusing. And we thought, of course, at first pass, maybe it has to do with land use history. It's a problem if actually the settlement hadn't happened and there were Gloeotrichia blooms. So we went and cored lakes in a number of different places, again with Holly at the lead, many of them in Maine, where she's based. And these data are hot off the press and about to be submitted. And all I want to show you here is these are the several lakes that we cored. And in this case, let's see, it's on your left. Nope, it's on your right is most recent time. And on your left it goes down to 1600. And the scale is different, but you can see that the Gloeotrichia abundances varied a lot and were, again, in some lakes higher in the 1800s, higher much earlier than today, and in some cases showed up today. So it's a conundrum, and we're looking at all kinds of factors that might have led to these blooms. So what about a little bit about the community dynamics? Kathy Cottingham and Cary are excellent community and population ecologists, as well as ecosystem scientists. And we started looking at the surface abundances, so how many of the colonies per liter are at the surface of the water at 4 different sites. One time it was 6 or 7 different sites, 4 different sites each summer. And what you can see here, and all I want you to see here, is that again, they're highly variable. From site to site and from year to year, these abundances vary. So a little bit about the ecology. Again, my interest and my passion has been about the biogeochemical end of things, but it's some of the community and population dynamics that clearly are the key to understanding that. So there's colony division in the water column. You get a formation of resting stages and senescence of this organism, overwintering and germination at the sediment. So we've also been looking at, and for some of the models that Meredith and Kathy have led, we've looked at recruitment too. So we need to know what the recruitment is coming up from the sediment. We've been monitoring this recruitment, again, since Cary was an undergraduate student from 2005 to present, mostly at the red dot sites. And it's this recruitment data set is completely unique. It's the longest of its kind. Most others are under 2 years, and we're trying to ask the question, what is it that drives interannual variability in recruitment? What is it that drives heterogeneity at all the different scales at which we're seeing it? And this is another place where citizen science sampling has been incredibly important. This citizen scientist, this is called the grandmother data set, and her grandchildren have been sampling daily of toes, doing sample toes daily since 2008. And so if we look at these daily toes, we get, again, heterogeneity. It varies widely within and among years in terms of what the estimates of Gleotrichia in the water column are. So here's where we are for one aspect of this project right now, which is, you won't be surprised, we're asking, what drives these different blooms? And so we have supplementary data, ice out dates. We don't have ice on dates, but we have ice out dates. We have water temperatures at high frequency. We've looked at stratification and mixing. The theory is that mixing is bad for cyanobacteria. These guys have gas vesicles, so they can float up to the top even with stratification. Precipitation, nutrient loading, and the challenging answer right now, through a lot of modeling and data analysis, is we can't do any better than predicting tomorrow's bloom based on today's data. So the next step for us will be actually to get a group of graduate students working with a Bayesian statistician, a colleague of mine, to try to see if we can take some Bayesian approaches to understand how it is we might better predict. So these forecasting these blooms, it's even with all these long-term data and these high-frequency data, this is a little bit challenging and depressing. There isn't a magic bullet yet for forecasting, and I've been interested to talk to a number of people here about some of the forecasting efforts, and this is one of those systems where we would like to be able to forecast, and we're having quite a few challenges. So it does seem like it's multi-factor, the causality, and it's context dependent. So again, this is using all kinds of data at different frequencies. So just a few things on the ecosystem effects as well. As I said, my interest is in the ecosystem effects and what we understand about this system. And the first is looking at productivity. So I'll talk in a few minutes about buoys and lakes. But based on a metabolism model, looking at high-frequency data and running those data through a model. Dave Richardson took the lead on this, who's at SUNY New Paltz. And looking at the patterns of photosynthesis and respiration, what we do see over time from Lake Sunapee is that there's more variability. And one question we have is it the Gleotrichia that's sending that signal? So we have a lot of other work that we're doing with some of this modeling, but I won't talk any more about that piece in this section. Okay, so here's the other question that I posed at the beginning of this talk is, can these species that actually fix nitrogen and have access to P, can they be internal drivers of nutrient cycling, and does it matter at their densities? So we know from eutrophic systems that this internal loading can be important. So about 2/3 of the internal loading was estimated for Green Lake in Washington some years ago, and in Lake Erkin in Sweden. So we know that it can be important in those systems. And here's what the phosphorus looks like for our estimates for both low-end estimates on your left and high-end estimates on the right, given the blooms and what we know about phosphorus and nitrogen in these organisms. And so, again, you can see that on the y-axis is the kilograms of phosphorus per season. You can see it varies a lot from basically an order of magnitude of what these organisms could contribute internally to loading. And by comparison, this is an estimate that was done, this is the only estimate actually that was done for Lake Sunapee for tributaries, looking at loading of phosphorus from tributaries in Lake Sunapee. And the number of tributaries is on the y-axis. And basically, our estimates are currently right about, well, they look like a small nutrient source, like a small tributary nutrient source. This high source drains a golf course, so that's the reason for that. If we had a tenfold increase, which is not impossible, we could add 10 kilograms of phosphorus to the current estimate. And if we had sustained recruitment, I mean, if the numbers keep going up, we could end up at a reasonable input of phosphorus relative to the external sources of phosphorus. So that led us to think a little bit about what this kind of internal loading might mean relative to external loading, and put together a kind of a simple model of nitrogen and phosphorus cycling in lakes. And it also has led us to think a little bit about resilience and thresholds. And resilience in this case, when I'm talking about that, I'm talking about the ability to remain in the same regime despite external forcing. So this is the kind of classic resilience diagram that you may have seen. And what we're asking about is whether there are some switching feedbacks in some of these deep lakes where you've got a clear water lake with low nutrients, turbid lake with high nutrients, and do you get a loss of nutrient limitation when you have an internal forcing, such as this? So again, if you're thinking about what contributes to internal nitrogen and phosphorus loading, hypolytic anoxia is one, the geochemical version. But we think, again, here it's cyanobacteria. It's actually a biologic mechanism that contributes to nitrogen and phosphorus that then contributes to eutrophication. And so we're, again, wondering whether this mechanism might make these systems less resilient. It'd be much harder to push them back to an oligotrophic state if they've got this internal mechanism that is recycling nitrogen and phosphorus. So the bottom lines from this part of the study is that they can fix N, they can access sediment phosphorus, they can access phosphorus that's available, whether lysed from them or from some other way. And so we actually really think that these cyanobacteria might be driving rather than riding this train. So again, if that's true, these cyanobacteria may decrease resilience, and that's kind of a significant problem in thinking about tipping points. We've seen actually in lab experiments, Cary's lab experiments, that these Gloeotrichia do increase nitrogen and phosphorus, and they change the community structure as well, which is subject of a different talk. So there are a bunch of different projects that are still going on and will continue to go on about cyanobacteria in Lake Sunapee. One I'm not going to talk about is a coupled natural human system project, which I didn't think to put in here, but after talking with a number of people, we may want to talk about it afterwards. This one is a recently funded NASA project where we'll use drones with a hyperspectral instrument. We'll use an app to collect data and use citizen scientists, engage with citizen scientists to monitor these blooms in clearwater lakes, deep clearwater lakes around northern New England. We're starting with Lake Sunapee. It just got funded, and we'll also consider what's going on or what has gone on in the watershed as well as drivers of some of these blooms, with the goal, ultimately, of being able to predict. There's a kind of a sub-goal or a primary goal also of building some of the algorithms that we need to build between the in-lake cyanobacterial bloom data and using remote sensing and the hyperspectral instrument. So I'm going to stay with Sunapee for a minute, but then branch out here. So going big scale, you probably also know that there are, well, last count, 117 million lakes, freshwater lakes on the planet. They cover a pretty small percentage of the Earth, maybe 5%, but they're disproportionately important in terms of our well-being. And while the Global Lake Ecological Observatory Network doesn't have a goal of trying to understand each one of these 117 million lakes, its goal is actually to understand lake function around the world. So when I was at Lake Sunapee for my sabbatical, I got asked to do a site review at the North Temperate Lakes in Wisconsin. And when I was there, one of the staff members said, he showed us a buoy and he said, we've got this GLEON project going. We're trying to have buoys all over the world, along with their scientists, and we're trying to understand lakes. And we're going to share data, and the world will change. And I thought, wow, that's a great idea. And I took that idea back to the citizen science group and said, boy, there's this global network where you can collect data, share data, and begin to interpret those data. You know, do we want to be part of this? And they said, we'll buy a buoy. We'll put a buoy in. We want to be part of this. And lo and behold, this is 2005, we found out that this GLEON network was great. It was 4 scientists, very creative scientists, a couple of research buoys, a concept, and importantly, a logo. So it was not plug and play. And that turned out to be actually a serendipitous benefit. So they were just getting started, getting this global network together. And I, as well as the Lake Sunapee Protective Association, have been integral to its growth over the last 12 years. And it's been a real opportunity to make sure that not only limnologists and computer scientists were involved, but actually citizen scientists, people who are organized and were interested in lake health have been involved. So it's been a terrific ride, and we've been able to help shape GLEON. And in fact, the Lake Sunapee's buoy was the first site for GLEON. So we have a mission now that is quite different from the original mission, which was about cyber infrastructure, and it was about getting buoys in lakes. And now it's mostly a group of scientists, but not entirely, to conduct science, basically, by sharing and interpreting high-resolution sensor data and also low-resolution data, so boats in the water data as well. And the goal really is to predict, to understand, to communicate how lakes function and are responding to a changing global environment. And what's really important about this network is that it's grassroots. So there is no top-down money. There is only a passion about doing science, doing good science, and doing it in groups. And I want to spend a couple of minutes kind of talking about how it is that we're organized, because I think it mirrors some of what I've heard that's very innovative that all of you are doing here. So we're doing network lake science. We're actually 3 networks. We're a network of lake observatories. We now have more than 150 lakes that are registered, 120 buoys. And again, this is not, no one is sending money to these places to install these buoys. About 40 observatories across 6 continents and 55 countries. And we're a network of data, and this is beautiful buoys around the world. This, for those of you who deal with big data, which means, in this case, heterogeneous data, this should scream heterogeneity and headache in terms of putting the data together. Our original goal, again, was plug and play. But different sensors, different frequencies, people call temperature a different thing across the world. So these are some of the things that get measured at all sites and are used in many of the analyses. So we're a network of data. We have probably 200 publications now and theses that have acknowledged GLEON. But most important, it's a network of people. Science doesn't do itself, people do science, and we learn this not really the hard way, but it just became very clear to us that making sure that people could do science as easily as possible and had the environment in which creativity could be brought to the fore is really important. So we have, again, a number of different countries, around 55 to 60, and really importantly, a third of this membership has been and continues to be early career and graduate students. And one of our National Science Foundation program officers said at one point that is an indication of sustainability. And so it's actually, it's turned into a network where students take a lot of leadership with those of us who are more gray, co-leading from within. So we've been asking ourselves kind of what keeps us together and what helps shape the science. And we've learned that face-to-face meetings are crucial. We have a lot of virtual meetings, but face-to-face meetings are crucial. We have a governance structure that evolves. We have working group activities, so our meetings are not meetings with a lot of plenaries. Our meetings are when we break up into groups and topical areas and produce, or at least get things going for producing papers or open source code. And we have this ethos of data sharing and collaboration, which wasn't, when we started, there really wasn't such a thing as team science, or at least it wasn't being talked about, I think, in our fields. But we're doing seat of the pants team science, collaborative science. And as I said, really importantly, GLEON Student Association grew up on its own, and they have taken a lot of leadership. And we also have a collaborative climate committee, which turned out to be incredibly important. At one of our first meetings, early on, all the senior people said, wow, what a great meeting. That was a fantastic meeting. And we happened to do a survey. And the early career people said, boy, that was terrible. You know, I ended up getting the coffee from my working group. Or, you know, I was talked over at the meeting. So we said, this is not our goal. And so the Collaborative Climate Committee actually helps us structure all of our meetings. And again, it's challenging because it's across cultures. It's not just, you know, lab-to-lab culture, it's country-to-country culture. And this Collaborative Climate Committee really helps us be productive. Just to give you an idea, we've certainly had Canadian participation. Actually, the chair of our Collaborative Climate Committee was from Canada, and again, it rotates around. Currently, Paul Hansen and I are the co-chairs of the GLEON Steering Committee. So again, to give you some idea of the topics that we address and the papers that are coming out, these are kind of the broad categories, although they're dynamic. I don't think we have an under ice group anymore, and maybe not a microbes group, and we do have a citizen science group. But this is the kind of structure that we break into for working group meetings. And again, it's diverse in terms of career stage especially, and country. And you know, since I've been talking about this, I think all of us in GLEON have been amazed at the importance of collaboration, understanding how to collaborate, how to do team science. And I thought this is really interesting. This is, again, our National Public Radio that came on last week. It said up to 70% of the tasks in most jobs are on track to be automated, leaving only the most creative, empathetic, technically fluent, and collaborative work for humans. And I think this is fast coming to science as well. It's pretty clear that those knotty questions that are interdisciplinary they require team science. As I said, we have face-to-face meetings, and I've just put red arrows near some things that were really important that happened at some of these meetings. And again, they just kind of happened grassroots style. One is the Graduate Student Association in 2007. Then we met with a lake association in New Zealand, which was kind of the beginning of us engaging with lake associations. A modeling working group was formed in 2010, and they have produced a number of open source models. Started out with the physics of lakes, and now are moving into biology as well. In Sunapee, New Hampshire, we co-hosted a meeting, and we had a public lake issue forum where we had people from around the world say in a few minutes, what are the issues, and what are scientists doing, and what might citizens do? And it was really, really quite interesting, you know, ranging from a woman from Ireland stood up and said, we have too many sheep in the watershed, and we've got phosphorus issues. A guy from New Zealand said, we have too many cows in the watershed. They've shifted from sheep, and we have nitrogen issues. And our colleague from China said, we've got people issues, and it's drought in some cases, and it's too many nutrients in others. And we've repeated that. We repeated it in Canada a few years ago, where we had a meeting in Quebec, and it's just quite remarkable. We initiated experiments across the world, doing spring blitz experiment measurements, and storm blitz measurements. And then we've been developing things like an app, a data quality app. So the research that has been enabled, and it's taken quite a long time actually, it probably took 6 or 7 years before we started seeing papers that had more than one lake in Wisconsin in them. So we now have there are 25 lakes and 40 lakes, and and quite a number of papers that include many more collaborators and more lakes. But these are the kinds of topics that we've been addressing as a group in GLEON, anywhere from episodic events to looking at carbon moving in lakes to kind of coupling and understanding when physical processes and biological processes are important and how to tease them apart. Again, open source model development, site-focused and comparative approaches, and large-scale experiments. And it's been really interesting talking, at least in the US and around the world, about how to sustain these kinds of networks in the absence of funding. And we've had some very interesting responses. In the US, I think they hold the GLEON group up as a great example of grassroots networks. I was asked to go to South Korea last year and talk to the group there, who also have an institute on global water and global water security. And it's kind of an anathema to think about these networks across political lines. So again, here's some of the research that's come out, and they move. We're now publishing more and more on how you form and manage collaborations as well as the science. And one of the things that's been really interesting is this is a network analysis that I've been working with some colleagues on. We started this a few years ago, so the publications were fewer. But we wanted to look at early career scientists with the hypothesis, we don't have the data to test it, but with the hypothesis that there are collaborations that happen as a result of this network that would not happen. Either they wouldn't ever happen, or they would happen much later in life for early career scientists. So often they're collaborating with people who are not their advisors, who are in their field and in a different country. And it's the purple is the early career scientists, and this is the strength of the connection, and the size has to do with the number of, or the connection with authors, the number of publications, and the number of connections. So here's one of the papers that I think is beginning to have a pretty high impact. That was led by Chris Solomon, who was a postdoc with GLEON, and it's had a pretty big impact with, at least in my experience, with citizen scientists as well. So if you talk to them about what's the use of having a buoy and a network of global scientists, and you say, well, we can measure or we can model gross primary production, photosynthesis around the world, and they kind of go, right, great. We don't have any context for that. But if you put it in context and you say, well, this is Lake Sunapee, and they see the pattern and they see the maximum, and this is, in contrast, Lake Taihu in China, and then kind of look at the difference between the productivity in Lake Taihu, China, and Lake Sunapee, it begins to, I think, for the research community, but also for the citizen science community, it gives a gradient and some sort of understanding about what the differences are in ecosystem function, and ultimately how you move from one end of the spectrum to the other, or hopefully not just one way. With this paper, we're also looking at autochthonous and allochthonous, inside, outside loading for respiration, and began looking outside of the lake. So this is, as a system scientist, I was one of the few people who was not a lake scientist at the beginning, and so there's a lot of looking at the lake, and much more now looking into the watershed and the airshed for this work. We were able to capitalize on a cyclone, the hurricane, that happened right before a meeting we had in 2011. And it happened that there were a number of people there who had buoys in the water, and we wanted to ask the question, how does the lake respond in real time to a hurricane? It was huge on the East Coast. And so here are the lakes, and you'll notice a couple of them are in Canada as well. And this is the storm track. The red is the storm track, and the blue is the circles. The colored circles are the maximum wind speed. And you can see actually the storm track didn't go right over the lakes, went close to the lakes. And so we looked at heat maps. We looked at how the storm mixed up the lake when the storm came through, and how long it took for the lakes to restratify. This is arranged by latitude, And you can see the lakes Canadian lakes down in the right-hand corner. And really importantly, part of this group of lakes is the New York City water supply. Upstate of New York City by about 120 kilometers are the reservoirs that are New York City water supply. And they have been this group has been part of GLEON for the last several years. They use buoys and they use models to make decisions about when they need to switch their sources. And this was one of the reservoirs that took the longest time to respond or to revert after the hurricane. And this is what it looked like for a very long time after Hurricane Irene. And the reason for this, of course, is the watershed. It's what's surrounding that reservoir. So it was quite remarkable to see the difference in response and the fact that it's not only intensity of the rain, but watershed clearly matters. With Paul Hansen, have led a GLEON fellowship program with National Science Foundation funding, where our goal is to take students from a number of different places and put them all together and do a research project, a big data research project, and also learn some leadership skills. And this is the paper that was led by Hilary Dugan, who was a postdoc, also Canadian, on this project. And it came out last spring looking at long-term data. This is not high-frequency data. But long-term data showing that where chloride is increasing, is a strong relationship with impervious surface in a buffer right around the lakes. And there's a lot of Canadian press. I don't know if it made it this far west or not, but in the east there was a lot of Canadian press for this study. And then finally, we have a group that again is led by graduate students who are looking at the role of carbon in lakes and carbon in the global carbon cycle, and kind of categorizing lakes by whether they hang on to carbon, pass carbon through, or emit carbon. So there have been a lot of partner networks that have sprung up, and is Lake Pulse the right name for the partner network that is right here in Canada? There's some recent funding, I think, that with whom we're working in GLEON. And all this is to say is that the power of networks and resulting in new networks and new collaborations, I think, is quite high and quite interesting. And then finally, we're setting off on our next 5 years. We do have some funding in order to bring people together. And that's the only kind of funding we've had for GLEON, other than the small research projects that have had funding. And this one is to expand further to global, but also include our colleagues who are working in atmospheric fluxes, in watershed fluxes, and to do a series of network experiments and expeditions. So that's where we're headed next. So again, what I wanted to do is to introduce you to some of the puzzles that we have regionally and locally, and also some of the ways in which I think some of these puzzles can be answered in the future, expanding networks to the globe, if not regionally. So I thought this actually, the other part is that I really am fond of the idea of thinking about where and how citizens might be involved in having their finger on the pulse of science and also contributing. It's not just a one-way arrow. It's not what we learn, we contribute to citizens. Here's what we know about lakes. It's more about citizens bringing to attention what some of the issues are, and oftentimes they have a really good inherent knowledge of systems and can point to potential ways of interpreting data as well. This is from Dave Schindler, actually, talking about citizens being really equipped to have their finger on the pulse. So I'm going to leave you with that and ask if you have questions.
All right, thanks, Kathy, for a really fascinating talk. Time for some questions. Any questions, comments? Maybe I'll start off with one. I guess I'm curious with your buoys. We face this in catchment science all the time. We make measurements in different ways, and it often thwarts intercomparison. So I'm wondering how you've overcome that, because I guess if you'd mandated measure these things in these ways, it would have benefits for intercomparison or the kind of big data analysis, but maybe it would then thwart uptake. I don't know.
Yeah, it would. In a perfect world, we would be able to do that, and we talked about having metadata standards and all kinds of standards. The problem is that many of these buoys had been in existence for research projects, and so people had been measuring for their purposes in ways that they measured. And so the huge job for many of the people who are leading these efforts, which are usually early career people, is to put together the data sets. And so we've gotten to the point now where people will say, well, I have this question about whether stratification is changing across the globe. Let us know if you want to collaborate and if you have data for thus and such a date and from thus and such a sensor. And so here's the time period, and then there's a template that goes out so that people can get their own data in that form. It's SneakerNet. Yeah.
That's great. Questions? Yeah. Graham.
I wonder if there's such thing as bloom weather.
Yeah.
Is there a particular kind of weather pattern that sets up blooms?
Yes. Yeah, it's a great question. From the statistical standpoint, we can't quite say that yet. But from observation, you kind of think, well, we have an early spring and a very warm beginning of summer. The Gloeotrichia bloom, they're holiday bloomers, 4th of July and Labor Day for us, our holidays. So we often see blooms earlier or stronger. I wish we knew the answer to that. Our colleagues in China actually put out a series of buoys on Lake Taihu, and they've been able to get some models to be able to predict 3 days in advance for some of their blooms. Different system. And maybe there's someone here. I mean, I know that there are a bunch of people who are working on ecological forecasting and bloom dynamics. Anybody here who has seen a
Well, some
predictor?
working in Regina, right, with the water supply there. Wondering Paul or Colin or Helen if she's here.
More focused on toxins than bloom dynamics.
Uh-huh. Yeah.
Do you see a difference between countries how grassroots support is manifesting itself?
Yeah. Well, support. To define support. What we do see across countries is sometimes there are barriers to sharing data. And sharing data and ideas are the basis of grassroots networks. So I would guess that if you were to go and count or look, that you would find in places where people don't want to share data, not much support for grassroots networks.
Okay, thank you.
I know there are a number of, I think it's a terrestrial network that, whose name I can't remember, in Australia, not looking at lakes, but looking at terrestrial systems, and has quite a bit of support. But
I guess I'm seeing Sunapee Lake with nice lake houses around it, and I'm imagining lakes in other places that are quite different, and I'm wondering, grassroots there, people have been at the lake for a long time, other places maybe a different approach. That's what I was getting at.
Depending on the communities that are surrounding them. Well, so the GLEON grassroots network really was started by scientists and is mostly about scientists. So I was thinking about that aspect of things. There are lake associations all over, well, it turns out all over the world, but all over this country. And actually, there were a number of people from lake associations who came to our meeting in Canada. And so that's the grassroots organization, and it's usually locally focused. And in this case, with the Sunapee group, I'm kind of impressed upon them that it's locally focused, but they're globally relevant for what it is that they're doing. But they're mostly interested in local.
Okay, thanks.
Good. Other comments, questions? Okay, they're eager to go to the bar. Graham?
Yeah.
I wonder if in GLEON, if the citizen science aspect has gone beyond just collection of data, but actually analysis and interpretation of data.
Yeah. I don't think so. The citizen science group per se is relatively new, the working group, and it was initiated in the Netherlands for one of the spin-off projects that I had on that big graph. And as far as I know, they are mostly collecting data on microbeads. They're collecting data on decomposition, and I don't know that they've been interpreting data. But for another project that I didn't talk about, a modeling project, this coupled natural human system project, we're building models, and one way in which we'll be doing interpretation is by playing with those models and doing scenarios, and that's kind of the beginning of getting analysis or doing analysis with citizen scientists. Yeah. You thinking about crowdsourcing kinds of things?
Yeah, just more like, yeah, so people might have all these different hypotheses about what's going on, and if we can bring them into the analysis, then they can sort of, some of those hypotheses can get, done away with, and new ones can emerge.
Yeah,
the more that they're actually involved in the analysis. And I just wonder if GLEON is doing that.
No, and GLEON really is about its members, so it's only limited by people's ideas. So, I mean, that would be a perfect kind of thing to, to come to a meeting or engage with GLEON and say, hey, I'm curious about this. We've done it verbally. We always sit down with the Lake Association when we have our science meetings and our data fests, and often they contribute in really significant ways to our interpretation. But I think it'd be really fun and really interesting to crowdsource in that way for some of the questions that we have.
I guess for me as a catchment hydrologist, a lot of the variability you're seeing, for me, would come back to catchment hydrology, what's feeding the lakes. And I'm wondering, within GLEON, are you able to look at that? I imagine the infrastructure's, challenging for getting all the inflows and outflows looked at physically, chemically,
Yes,
as opposed to buoys in the lake, which maybe is more tractable from a citizen science point of view. How are you addressing that part of it?
Well, as I said, from the beginning, it was mostly a very lake-focused group, and we now have a watershed working group, and so there are lakes and watersheds that have those extra data, and they are being incorporated into a data set to ask those kinds of questions about the watershed. So I think there's a huge awareness that we need to turn some attention there, and also a big lack of data.
Right. Good.
Yeah.
Well, I think we'll transition now, for those that are interested, to Boffins for a whiskey or a Coke or a beer, and we can continue the discussion about lakes. But please join with me in thanking Kathy for a really stimulating seminar. Thanks.
Thanks, Jeff.